DeepMind's AI makes gigantic leap in solving protein structures. Hello futurists. Welcome to my YouTube channel, where I track the acceleration of intelligence that will inevitably help us build a brain. In today's video, we're going to look at how DeepMind, the subsidiary specializing in artificial intelligence, beat the best groups in the world in a competition aimed at predicting the 3D structure of proteins. The subsidiary specializing in artificial intelligence beat the best groups in the world in a competition aimed at predicting the 3D structure of proteins. DeepMind, the subsidiary of Alphabet, the parent company of Google, specializing in artificial intelligence, gained fame in 2016 when it achieved the feat of beating the world champion of Go, an Asian strategy game, with its AlphaGo program. The following year, DeepMind got even stronger with AlphaGo, a program that learned the game of Go by playing games against itself, which allowed it to crush the previous champion, AlphaGo. After these two demonstrations of strength, DeepMind and its founder, Demis Hassabis, tackled a whole different challenge: protein folding. A fundamental problem in molecular biology which has held researchers in check for several decades, and whose solution would be nothing less than a revolution in biology, both for the understanding of living things and for the production of targeted drugs. And this first test for the small British structure was an impressive success. In 2020, DeepMind has brilliantly won the benchmark international competition, CASP, or Critical Assessment of Structure Prediction, far ahead of the best academic teams on the planet, some of which have been working for many long years on the subject. In CASP, DeepMind's program, termed Alphafold, surpassed about 100 other teams. In 2018, the first year that London-based DeepMind competed in the CASP, Alphafold came out on top. However, in 2020, the company's deep learning network outperformed all other teams and, according to experts, performed so well that it might usher in a biological revolution. Proteins are the fundamental building blocks of life, and they are responsible for the vast majority of events that take place within cells. Proteins have a tendency to take on their final structure on their own, governed exclusively by the rules of physics. Proteins, and how they move and change, are involved in nearly every function that our body performs, such as contracting muscles, sensing light, and converting food into energy. The unique 3D structure of each protein determines what it can do. Antibody proteins used by our immune systems, for example, are Y-shaped and form unique hooks. These antibody proteins can detect and tag disease-causing microorganisms for elimination by latching onto viruses and bacteria. The 3D form of a protein determines how it functions and what it accomplishes. The axiom of molecular biology states that structure determines function. For decades, laboratory procedures have been the primary method of obtaining high-quality protein crystal structures. Beginning in the 1950s, the first full structures of proteins were determined using an X-ray method, in which X-ray beams are fired onto crystallized proteins, and the diffracted light is translated into the atomic coordinates of the protein. The majority of protein structures have been discovered through the use of X-ray crystallography or cryogenic electron microscopy, where samples are cooled to cryogenic temperatures and embedded in an environment of vitreous water. Physicists have long puzzled over how a protein's fundamental pieces, a string of various amino acids, map out the many twists and folds that eventually define its final structure. Because there are more interactions between amino acids to consider, the larger the protein, the more difficult it is to model. According to Levinthal's paradox, randomly enumerating all possible configurations of a typical protein before reaching the true 3D structure would take longer than the age of the known universe. But proteins fold spontaneously in milliseconds. The protein folding problem, which scientists have been working on for decades, is predicting how these chains will fold into the complex 3D structure of a protein. According to the researchers, early attempts to utilize computers to predict protein structures in the 1980s and 1990s were a failure due to poor performance. When other scientists used the methods described in published publications to different proteins, the lofty promises made for them tended to fall apart. M. I. Kobayashi, a computational biologist at Columbia University in New York City, who is also taking part in the CASP contest, is looking forward to delving deeper into the details of Alphafold's performance. It's possible, but doubtful, according to him, that a crop of protein targets that was simpler to grow than normal contributed to the performance. Kobayashi has a strong suspicion that Alphafold will have a transformative effect. He says, "I believe it is safe to state that this will have a significant impact on the area of protein structure prediction. I believe a significant number of people will quit the profession because the underlying challenge has arguably been solved," he adds. In the words of the Nobel laureate, "It's a breakthrough of the first kind, and unquestionably one of the most significant scientific findings of my lifetime." So, why this work on proteins? Well, proteins are the workers essential to the functioning of all living things, from bacteria to complex organs like the human body. The recipe for producing them is encoded in DNA, called genes. They have extremely varied functions, from making muscles work, converting food into energy, or even recognizing an unwanted virus in the body. However, these functions are mainly governed by their form, their three-dimensional structure. Scientists try to determine the structure to understand how a protein works. But there is a catch, a big catch. Because the DNA sequence linked to a protein actually gives rise to a large linear chain, a long necklace whose beads are amino acids. Some of these beads are hydrophilic or hydrophobic, others have positive or negative charges. Properties which mean that, according to the succession of the different pearls, the long necklace that is the protein will fold up on itself to result in a particular three-dimensional shape. Yet, despite decades of effort, scientists still have a hard time predicting this 3D structure from the amino acid sequence. The most powerful supercomputers on the planet cannot calculate all the possible aliasing combinations. Today, the only reliable methods of knowing the structures of proteins are experimental. This requires expensive equipment to observe the shape of proteins, X-ray crystallography systems, nuclear magnetic resonance, and more recently, very high-resolution electron microscopy. Techniques are progressing, but the characterization of proteins remains long and expensive. In some cases, for sample reasons, you can't have enough precision to have an atomic resolution, says Michael Nigels. This is where DeepMind comes in. Its Alphafold program crushed the competition at the 13th edition of the biannual CASP competition. In the most difficult category, predicting the shape of a protein simply from its composition, without any additional data, DeepMind topped the competition for 25 of the 43 target proteins. The runner-up only made the best prediction in 3 cases. Scientists have long been curious about how a protein's fundamental pieces, a string of various amino acids, map out the myriad twists and folds that make up its final structure. Researchers claim that early attempts to predict protein architectures using computers in the 1980s and 1990s failed miserably. When other scientists used the methodology to different proteins, the lofty promises in published studies tended to fall apart. This is why CASP was created in 1994 by John Malt, a computational biologist at the University of Maryland in College Park, with the goal of improving computer methods for reliably predicting protein structures. The competition requires teams to predict the structures of proteins that have been solved using experimental methods but have not yet been published. Malt credited the experiment, which he doesn't refer to as a competition, with substantially enhancing the field by putting a stop to overblown promises. He explains, "You're actually figuring out what looks promising, what works, and what you should avoid." DeepMind co-founder and CEO, Demis Hassabis, said the business aims to make Alphafold usable so that other scientists may utilize it. It had already released enough information about the initial version of Alphafold to allow other scientists to reproduce the method. Alphafold can take days to generate a projected structure, which includes estimations of the protein's reliability in various areas. "We're just getting a sense of what biologists want," Hassabis says, citing drug discovery and protein design as examples of possible uses. In any case, DeepMind's approach is original. Competitive teams that also use artificial intelligence systems have often performed machine learning by going to databases of tens of thousands of already known 3D protein structures. Michael Nigels says, Alphafold proceeded in a different way, using 2 other methods, one which assembles the structures of fragments of a few amino acids, and the other which analyzes variations in sequences of similar proteins to infer contacts between amino acids. No one knows the exact secret of DeepMind's success, but they have a great deal of expertise in neural networks. They also have the infrastructure to do these calculations, and finally, they have found the right data for learning. Many experts in the area were taken aback by DeepMind's success at CASP 13 in 2018, which had hitherto been the domain of modest academic organizations. But according to Jinbo Zhu, a computational biologist at the University of Chicago in Illinois, the team's technique was generally comparable to that of other AI teams. The original version of Alphafold used structural and genetic data to estimate the distance between pairs of amino acids in a protein, using the AI technology known as deep learning. According to John Jumper of DeepMind, who is driving the research, Alphafold uses this information in a second stage that does not use AI to come up with a consensus picture of what the protein should look like. The team sought to expand on that strategy, but finally ran into a brick wall. So it took a different approach, according to Jumper, and created an AI network that included more knowledge about the physical and geometric restrictions that influence how a protein folds. They also gave it a tougher task. Instead of predicting amino acid associations, the network now has to predict the final structure of a target protein sequence. It's a much more complicated system, Jumper explains. So, currently, Alphafold is unlikely to close labs that employ experimental approaches to solve protein structures. However, it's possible that low-quality, easier-to-collect experimental data will suffice to produce a decent structure. Because the tsunami of accessible genetic data can now be reliably translated into structures, some applications, such as protein evolutionary research, are destined to thrive. A new generation of molecular biologists will be able to pose more complex questions as a result of this. André Lupas, an evolutionary biologist at the Max Planck Institute for Developmental Biology in Tübingen, Germany, says, "It will necessitate more thought and less pipetting." CASP occurs over a period of time. Teams have several weeks to submit their structural predictions after target proteins or sections of proteins called domains, around 100 in total, are provided on a regular basis. The predictions are then evaluated by a group of impartial scientists using criteria that measure how similar a predicted protein is to the experimentally determined structure. The examiners have no idea who is making a forecast. The forecasts of Alphafold arrived under the moniker Group 427, but the surprising accuracy of several of them made them stand out. The majority of people had guessed Alphafold. Some predictions were better than others, but roughly 2/3 were on level with experimental structures in terms of quality. According to Molt, in several circumstances it was unclear if the gap between Alphafold's predictions and the experimental outcome was due to a prediction mistake or an experimental artifact. Alphafold's predictions did not match experimental structures measured by nuclear magnetic resonance spectroscopy, although this might be due to how raw data is transformed into a model, according to Molt. Individual structures in protein complexes, or groups, are especially difficult to model because interactions with other proteins change their forms. Overall, teams predicted structures more accurately this year than the previous CASP, but Molt attributes much of the improvement to Alphafold. According to Molt, other teams' greatest performances on fairly tough protein targets usually scored 75 on a 100-point scale of prediction accuracy, whereas Alphafold scored about 90 on the same targets. According to Molt, around half of the teams referenced deep learning in the abstract describing their technique, indicating that AI is having a wide influence on the sector. The majority of the participants were academic teams. However, Microsoft and Tencent, a Chinese technology giant, also participated in CASP 14. This is a big advance, but it is a bit misleading, comments Guillaume Pages, doctoral student at INRIA Grenoble-Rhône-Alpes, who took part in the competition in the team led by CNRS researcher Sergei Grudinin. In fact, the results of DeepMind vary a lot depending on the protein studied. If the target is easy, it does not do better than the others, sometimes worse. If it is difficult, it is often much better. Yet academics are sometimes very close to DeepMind. In the opinion of all specialists, the performance of DeepMind is very impressive, especially for a newcomer to the field. But that does not mean that the scientists at DeepMind have finally solved the problem of folding all the proteins, says Andrei Kajava, head of the structural bioinformatics team at the Montpellier Cell Biology Research Center, CNRS. There is still a lot of progress to be made, and it will be necessary to see in the future whether DeepMind still has a lot of room for improvement. The victory of DeepMind is an important advance, which proves that artificial intelligence and big data are more competitive than approaches based on physical models of folding, said Sergey Grudinin, present in Mexico, where the explanations of researchers of DeepMind were followed with great interest by all experts. With its own artificial intelligence software, Sergey Grudinin's team obtained good results in the CASP-13 competition, particularly in a category where it is necessary to predict the shape of the protein with a little more information than the chain of acids alone. Janet Thornt, a structural biologist at the European Molecular Biology Laboratory, European Bioinformatics Institute in Hingston, UK, and a past CASP assessor, says, "This is a challenge that I was beginning to fear would not get addressed in my lifetime." She expects that by utilizing this method, she will be able to better understand the function of the hundreds of unidentified proteins in the human genome, as well as disease-causing gene variants that differ across people. The success of Alphafold is also a watershed moment for DeepMind. The business is most known for using artificial intelligence to master games like Go, but its long-term objective is to create systems with broad, human-like intelligence. One of the most essential uses of AI, according to Hassabis, is tackling huge scientific difficulties like protein structure prediction. In terms of real-world impact, I believe it is the most significant thing we've done. The business revealed predictions of the structures of a few SARS-CoV-2 proteins that hadn't yet been identified experimentally in early 2020. According to Stephen Brohorn, a molecular neurobiologist at the University of California, Berkeley, whose team disclosed the structure in June, DeepMind's predictions for a protein called ORF3A ended up being remarkably similar to one later discovered by cryo-EM. What they've accomplished is quite remarkable. Alphafold code used at CASP-13 is available on GitHub, which I'll link in the description below for anyone interested in learning more or replicating their results. While there's reason to be ecstatic about the protein folding model's success, there's still a lot of work to be done in the field of protein biology. With the goal of making a real-world impact, it'll be exciting to see how AI will contribute to basic scientific discovery. This approach may help scientists better understand the body and how it works in the long run, allowing them to more effectively target and design new, effective cures for diseases. Only about half of the proteins produced by human cells have been mapped by scientists. Some rare diseases are caused by mutations in a single gene, resulting in a malformed protein that can have serious consequences for an organism's health. A tool like Alphafold could aid rare disease researchers in quickly and cost-effectively predicting the shape of a protein of interest. This method may eventually help us contribute to efficient drug discovery while also reducing the costs associated with experimentation as scientists gain more knowledge about the shapes of proteins and how they operate through simulations and models. Let's hope that AI will be useful in disease research and, as a result, will improve the lives of millions of patients around the world. However, the potential benefits aren't limited to health. Understanding protein folding will aid in protein design, which could lead to a slew of new possibilities. For example, advances in biodegradable enzymes, which can be enabled by protein design, could aid in the management of pollutants such as plastic and oil, allowing us to break down waste in more environmentally friendly ways. In fact, scientists have already begun engineering bacteria to secrete proteins that will help waste decompose more quickly and easily. This first foray into protein folding was a success, demonstrating how machine learning systems can integrate multiple sources of data to help scientists quickly come up with creative solutions to complex problems. And maybe, one day, just as we've seen how AI can help people master complex games through systems like AlphaGo and AlphaZero, AI breakthroughs will also help us advance our understanding of fundamental scientific problems. Even though there is much more work to be done before we can have a quantifiable impact on diseases, waste management, and other issues, we know the potential is enormous. This wraps up today's video. Thank you guys for watching. If you enjoyed the video, hit like, and if you want more content like this, subscribe to the channel. Make today a great day. Until next time, keep thinking.